Welcome to our policy event. The focus is really on strategies to combat antimicrobial resistance in aquatic ecosystems. Well, what can you expect today? So I will give you a short welcome and introduction on aquatic pollutants, the ERA-NET Cofund, and the funding measure as such. Then we will have a brief introduction of our policy brief. So until 1.5 hours ago, it was intended that Caroline from ISO will give this presentation. She just gave me a call that due to severe and very unpleasant family issues, she will not be able to join us today. So please apologies for that. So I will give the presentation instead of her. And we also had planned to have here a member of the European Parliament Water Group to join us and to give us a little overview on the actual policy situation that we have on the European level, but unfortunately due to short-dated commitments, so the chair and the two co-chairs had not been able to join us. But what we will do definitely is, after this policy event, we will, let's say, based also on the things we have discussed here and additional input that we might have gathered from this meeting today, we will finalize the policy brief, and the policy brief will then be also made public on our Aquatic Pollutants website, and it will be also handed over and sent to the members of European Parliament Water Group. I don't know if you're aware of this group. So this is an interest group of European Parliament members that have a special focus on water, so that are taking care as a cross-cutting issue on water aspects in their EU parliamentarian work. So for sure, with this policy brief, we will have a quite wide outreach. And in addition, we will also bring it to attention to the colleagues of the Water Europe network with their community of practices, so that we have a quite wide outreach also of this policy brief. Well, to continue with what you can expect, after the short introduction of the policy brief, we will do a little deep dive into the most important key messages of the policy brief that will be given by Carl Andre from ERTA in Spain, from Andreas Thiem here from Germany, from TSW, and from Victor Matamoros from IDEA CSIC in Spain. And we'll then have a joint discussion, and you as the auditorium have the opportunity to join us in these discussions and to bring in your input, your discussions on the issues, on the aspects that have been presented. Well, now let me start with a little introduction on the ERA-NET Cofund on aquatic pollutants. As you can see, it's running close to the end. So we as a networking and transfer project started in June 21, and we will end by the end of this month. We were one of the in total 19 projects, plus in principle the 18 research projects that have been funded in total by 32 ministries, authorities, and funding organizations from 26 countries. The call secretariat for this call was here related in Germany at PTKA. So that was a real pleasure. And that's also great, Mr. Müssner, that you're joining the meeting here today, because for those of you who don't know him, Rainer Müssner is in our Ministry of Research, Technology and Space. I hope I got that right, as we got a renaming a few weeks ago. He is the representative and the responsible for the water issues, and we are very happy that we had this funding measure in the ERA-NET Cofund and under the lead of the German Projektträger. Well, what was it all about? So the goals were really to determine pollution pathways of CECs, pathogens, and AMR, as well as their transformation behavior. So that means also a lot of understanding what's going on, what is there with all these substances. Then the assessment thing around risk management. So what strategies do we have? What can we do against them? And also, let's say, solutions. That means strategies and technologies to reduce pollutants, but also, let's say, to see if we can get a wider impact. So that means also to encourage policy change. That's also part why we have this webinar today. And let's say from the network and transfer side, we had a little deep dive. We were looking at what are key stakeholders, what are their knowledge gaps, their knowledge demands, how do they acquire knowledge, how do they acquire information that is important for their day-to-day business, for their decision making. So that was also a big part of our work. And of course, to create synergies amongst the 18 research projects, to set the collaboration with the stakeholders and to support the transfer of results into practice. So that's also why we have this policy webinar today, and why the next steps are those that I've already quickly lined out for you, what we will make out of this policy brief. Well, when we look at the aquatic pollutants ecosystems, as we have said, it's micropollutants, it's pathogens and antimicrobial resistance. This is a kind of an environmental map where the 18 projects of aquatic pollutants are located. The things they are addressing, you can see it's really quite wide. It's, let's say, from the marine and aquaculture sector to industrial production over various application fields, transports, pathways, impacts on surface waters, drinking waters, aquaculture, human health, ecosystems, and of course also the risk assessment, the monitoring, so all the stuff. And having in mind such a complex system, it was clear that the aquatic pollutants ecosystem is set in 3 elements. One was the focus on measuring, and you can see here on a kind of a little heat map which of the 18 projects were addressing which elements in this ecosystem, in the themes of measuring on the upper left side. Then, of course, evaluation. So we have information, something is there. So in the evaluation, what does it mean? And in the third field, of course, taking action to come to a reduction or avoidance of the occurrence or spreading of micropollutants, pathogens, and antimicrobial resistance in the aquatic ecosystem. So this is a little bit, in a nutshell, the overall picture where the aquatic pollutants projects were working, and where they have elaborated their results. And what we will do now today with you, we have already had a wide range of activities regarding the results of the projects. We had the final event on that. And today we want to focus on a set of recommendations to policy. And this is what the policy webinar is about. So that means we want to give you an insight in the key messages of our aquatic pollutants policy brief and initiate an introduction with you. This also means we will have a little deep dive in key findings from 3 aquatic pollutants projects regarding AMR, which stand, let's say, as kind of guiding lights for the others for a certain range of key findings that we have. It's findings that several projects had, but we've selected here for this webinar these 3 key findings. We'll give an insight in potential measures and policy actions and discuss them with you. And of course, get into the discussion how we can support future policies at EU levels based on the results that we have here in these projects and in this policy brief. Well, that's it already from my side with this quick introduction, a little bit setting the scene on the Aquatic Pollutants ERA-NET Cofund with its 18 funded projects with Aquatic Pollutants Transit, other networking and transfer project. So that means I'll take you now as a replacement for Caroline into the overview of the preliminary policy brief results, which will then be later on be described in more detail and with examples from the 3 presentations. All right. So therefore, I'm now sharing the next presentation with you. So here we are. As I said, so we have at the title for our policy brief, Safeguarding Water, Protecting Health, Policy Actions to Combat Antimicrobial Resistance. And just to take you on board, let's say what's about the policy brief, what's behind it. So the policy brief was developed within the Aquatic Pollutants Transnet project And the results of that, so the content is based on the findings of the 18 research projects that were funded under the ERA-NET Cofund. We have selected from a quite wide range of the projects. We have been able to bring in aspects that are relevant for policy issues related to AMR. And so that means we distill key cross-cutting issues and policy messages on AMR out of these projects from an environmental and a water perspective. And after several rounds of internal discussions and shaping and focusing on major issues, we derived with a set of 4 recommendations intended to inform discussions around environmental and health policy at the EU level. And this is also, again, what the webinar is about, and I'll give you a little introduction on these 4 things. First of all, of course, the question, as you have heard, we had this quite wide range of issues or topics in aquatic pollutants, micropollutants, pathogens, and AMR. And of course, the first question you might ask as well, why do you select AMR as an environmental challenge? It's quite simple because, as you might be aware, so the micropollutant issue we are all aware of already since years, partly also since more than 10 years. So, for example, on a national level in Germany, also from our Ministry of Research, Technology and Space, we had already in the early 2010s a funding measure that was dealing with micropollutants. And during this funding program, already in the late 2010s, the issue of antimicrobial resistance came up. And over the past years, it strongly developed. And we also saw in the aquatic pollutants projects that's a real hot topic. And when screening the actual ongoing developments on the European policy landscape, we have seen that the antimicrobial resistance and approach is one that could be stronger addressed to protect the aquatic ecosystem. Because what we see is, let's say, we are all aware AMR is often seen as a clinical or hospital issue. You all know these resistances in the hospitals, so the issues, the fight with the antibiotics against the gram-positive and negative bacteria, and the issue that we are running into a shortness of really powerful antibiotics to combat with this resistancy genes. But what's coming up, or we're getting aware over the past years more and more that the resistant bacteria and genes are widespread in the environment, and that we have a wide variety of pathways. And there, water plays a central role as it's a major, let's say, spreading pathway for AMR from anthropogenic sources into ecosystems. And as I already mentioned, so the current EU water environmental policies do not adequately address AMR. So we've seen from here a real challenge, a big issue is that it would be important with this policy brief to bring that issue stronger up. And the One Health approach is something that is already going on the European level, but it was important for us now to stress that tackling AMR really also acquires this One Health approach, really integrating human, animal, and environmental health as, let's say, the connection between all 3 compartments and all 3 actors is so essential due to this, let's say, distribution pathway over via water systems. Well, to get you a little insight into the 4 main recommendations, starting with the action one here. First, upgrading advanced wastewater treatment to support nature-based solutions to effectively target AMR, as I said. So if we can avoid the spread of resistant bacteria, resistant genes, let's say, within the wastewater treatment systems, it's more or less an avoidance at the source. And we know that there's a wide range of technologies that can help us with this. The advanced treatment technologies show very high potential to reduce contaminants. We see that, for example, in Germany with the advanced treatment step, that's mainly for the micropollutants where we're very successful. But we also see, let's say, also on the European level, that we have nature-based solutions that, especially on the perspective on hygienic aspects, are very efficient and well suited to reduce the spreading of AMR and AR genes, for example, from agricultural runoff or from stormwater runoff. So these are well-suited technologies there. And of course, it's nice to have these technical solutions, but we also need, let's say, removal targets that can be set so that we have also an idea where to head to with these technologies to have them implemented. The second thing where we put a spot on is taking us away, let's say, from the freshwater ecosystems more to the coastal and marine ecosystem. It's about, let's say, regulate international oyster transfer practices and update aquaculture setting requirements in addition to regulating antimicrobial use in aquaculture. So we have the oysters here because it was addressed in one of the aquatic pollutants projects. So it might also be an issue related, let's say, everywhere where we would see, and maybe Carl might address that also in his introduction. This might also be true, let's say, for aquaculture organisms that are hosted in coastal ecosystems and are transferred between countries in a trading process and being inserted again in new aquatic ecosystems. So these are, of course, potential transfer pathways to spread AMR. So this is for sure something that also we will then hear in one of the next presentations. For us, personally, for me, coming from the freshwater side, so this was a very interesting aspect, giving a new focus from my freshwater side to the marine and to the coastal side to see the mechanisms of spreading AMR there. So the third one is then that brings us also in the monitoring, and also with a relation a little bit helps us in the assessment thing. It's, let's say, establish EU-wide harmonized environmental AMR monitoring standards, because that's something that's not there at the moment. So the technologies to monitor and identify resistancy genes and bacteria have strongly developed in the past years, and so it's now the technologies are there, and now it's the time that we need a harmonized and integrated surveillance framework. And this is, of course, essential to have a clear idea: where do we have hotspots, where do we have real, let's say, transfer pathways, where do we have developments and increasing resistant GC occurrences, so where we can, let's say, have the basic tools that allow us to assess and properly manage all the actions and activities we are taking or we are planning towards avoiding the spreading of AMR. That also means expanding microbial resource tracking, and also, let's say, again here the link to the One Health goals to establish an integrated environmental surveillance with human and animal health aspects, to really have also the interaction or the interface between the different compartments that are covered in the One Health approach. So as you can see, let's say we have here three recommendations from the let's say detection monitoring side, from the, let's say, non-technical action side, risk management, risk reduction, and let's say with the nature-based solutions also, let's say, from the taking action side. But what we have in addition is also the issue on strengthening communication, improve knowledge transfer to enhance public awareness. That's a recommendation that we developed out of our networking and transfer project, because we have really seen we have various stakeholders. When we look to this One Health approach and to the wide range of ecosystems that are involved in all these things, we have a wide range of actors and stakeholders being involved in this One Health approach. They have different information sources, and we need to address them properly. So this is why we have the fourth recommendation in there. So to conclude, so the AMR, we have addressed them as their key environmental challenge. It's definitely requires an integration into the European environmental health environmental and health policies, also in the One Health approach. And so the policy framework needs to be coherent to allow us to, let's say, effectively address the antimicrobial resistance issue. Well, with this, that's from my side the quick introduction that I could give you on behalf of Karen to get you a little bit some first insights on the aquatic pollutants policy brief. And I would like to ask you now, let's say from your side, if there are any quick questions, understanding questions. Otherwise, I would kindly ask you to note down your questions and bring them into the joint discussion after we had these impulses in the introduction. So if there are no, let's say, direct urgent issues from your side, then we can move forward in our agenda. So we start with our first deep dive, the environmental spread and persistence of AMR in oyster aquaculture. And for that, I would like to hand over to Carl Andre from the Spare Sea project. So Carl, the floor is yours.
I'd like to thank Transnet for allowing me the opportunity to share this on behalf of our project coordinator, Matthias Wegner, and our partners in France and Italy. And as Thomas has already said, that a lot of this is coming from the perspective of One Health, and Spare Sea is no exception. Conceptually, it's coming from the perspective of One Health, where we're looking at the environment, humans, and also animals. And in this case, the animal is oysters. The oysters are given an emphasis in this context because they exist at the interface between the land and the sea, and also because oysters are something that are usually eaten raw, so they are a very clear bridge for transmission of live bacteria containing antibiotic resistance genes, from transmission of those from oysters to humans. And so for this, we had started out with, in the area of Boone, there's a hatchery for oysters, part of a collaboration with the CNRS, and the hatchery produced oysters with distinct environment antibiotic exposure backgrounds, but they're raised in a hatchery, so they're relatively clean. And these oysters were then partitioned and distributed to Sylt in the north of Germany, and in Taal Lagoon, and here in the Ebro Delta, in areas that have distinct environments, where in each place there's oysters being harvested for human consumption. In the case of Sylt, they're wild oysters, and this is an environment that's considered relatively pristine, whereas in Taal Lagoon and the Ebro Delta, there's a fair amount of agriculture and terrestrial runoff with agrochemicals. In the case of Taal Lagoon, it's vineyards, and in the case of Ebro Delta, it's rice culture. And so these distinctly different habitats were provided with these oysters from a common source that should be relatively clean, and so they were screened for infectious diseases before being transported to each of these three sites. Now, in addition to using these oysters, the SPF, specific pathogen-free oysters, for these field studies at each site, they were also used for a bacterial challenge to look at the immune response of the different cohorts of oysters, given that they had distinctly different antibiotic exposure histories. So there was a bacterial challenge done as well to look at the immune response of the oysters. In work package 1, there's 4 work packages. Work package 1 was looking at what's there and what's active, and for this we wanted to approach this with in various ways, using culture independent and culture dependent protocols. So the oysters, the SPF oysters, are not considered here in work package 1. These are commercial oysters from each site, and we collected oysters, prepared tissue homogenates, and put it on the oyster homogenates onto different media, either TCBS, which selects for Vibrios, or marine auger, and these contained different antibiotic selections. And then we incubated replicates of these plates at either 20 degrees or 37 degrees to get a chance to look at the environmental isolates versus the potential zoonotic pathogens that might be harbored in the oyster microbiome. Work packages 2 and 3, they were, we're missing, there we go. Work packages 2 and 3 were looking at the synergies involved in the accumulation of antibiotic resistance genes due to co-exposure to different kinds of agrochemicals in each environment. And included in this agrochemical topic was also looking at heavy metals. So and the work package 2 is mostly field studies, whereas in work package 3 is more experimental studies, where we try to reproduce some of the effects of the environment, but in a more limited, focused way. And as I mentioned, the work package 2, we used these SPF oysters that had just, they're coming from broodstock, and the broodstock either had chloramphenicol exposure or no antibiotic exposure. And the larvae or oyster juveniles from these two broodstock groups were then subdivided into three F1 larva groups, and they were given additional different antibiotic treatments. So we ended up with 6 cohorts of oysters that were then sent to the 3 sites, as previously mentioned, and they were exposed for either 2 weeks or 6 months. So these would be the samples that we talk about later, T4 samples or T6 samples. Okay, and so all of this was designed to identify what might be some of the environmental drivers. Work package 4 was looking at environmental compartments to see how the different environmental compartments were linked, what kinds of antibiotic resistance genes did they have in common, and how that might be linked to sources and sinks of specific antibiotic resistance genes. And we want to know how all of this contributes to ecosystem health. What are the anthropogenic activities that are contributing to the emergence of novel pathogens? Because some of the antibiotic resistance genes may be mobile and be able to be transferred from a non-pathogenic bacteria to a potentially pathogenic, human pathogenic bacteria. So in work package 2, one of the things we found initially in work package 1 was that the commercial oysters, they all had colistin resistance. Well, I should say, at all sites, we encountered oysters with colistin resistance. That isn't to say that all had colistin resistance, but we found oysters that harbored bacteria that had colistin resistance at all sites. And so this became interesting and more of a focus, because colistin is one of these antibiotics of last resort when you have multi-drug resistant infections. So we, in categorizing the colistin resistance that were isolated on the different kinds of bacteria, either TCBS, the left panel, or merino agar, the right panel, we further subcategorized them into those grown at 37 or 20 degrees. And if you look at the left panel, you can see that the gray, the light gray, is 37 degree cultures that were grown from each site. And you see that 100% of the cultures from Tau Lagoon grown at 37 degrees were also colistin resistant, whereas in Sult, where the water is much colder, there were no isolates that were colistin resistant and also grew at 37 degrees. So this is probably just reflecting a temperature gradient across sites, and whereas in respect to the other characteristics shown here, they seem to be quite site specific. When we look a little deeper in the colistin resistance, we see that there's, if we take the genes for the colistin resistant markers that we found in the metagenome sequencing, and we take those genes and convert it to amino acid sequences and then do a phylogenetic analysis of that, those amino acid sequences, the colistin resistance markers can be subdivided into two groups, either MCR, which is mobile colistin resistance, or EPTA. And this gold arrow in the center marks the branch point at which these two classes of colistin resistance partition. In the case of the EPTA, there was a distinct Mediterranean clade that was represented resistance markers that were only found in the Toad Lagoon and Ebro Delta. And then with regard to the MCR, there was also some degree of geographic segregation, but with more admixture with the gene variants that were found in Sult mixing together with gene variants from the Mediterranean sites. And the geographic site is indicated by the blue, green, or red rectangles in the outer circle. All of the gray and dark gray in the center ring are marking variants that were identified as part of this study. So there were 17 distinct EPTA MCR sequences, 15 novel MCR variants, and we also saw some similar geographic segregation among the class A beta-lactamase markers. In work package 3, we had a mesocosm experiment where we tried to parse out some of the environmental factors and look at them individually by looking at the kind of shellfish species in each tank, either mussels or oysters, or the density of the shellfish present, or the temperature, either 18 degrees or 23 degrees, or the type of sediment in which the shellfish were harbored, which was either sand or mud. And then we also had tanks where we contrasted these different variables together. And what we found was that the uptake of colistin resistance was more receptive to the majority of the variables tested, or combination of variables, whereas in the case of ampicillin and colistin uptake, they seem to uptake these resistance markers when they were cultured in the presence of sand rather than mud. In work package 4, we looked again at the environmental compartments, and here we're looking at the abiotic samples that were collected, water and sediment, and we've looking at subgroups, categories of antibiotic resistance markers that can be specifically associated to certain kinds of sources of waste, in this case hospital waste or aquaculture waste. The carbapenems are extended spectrum beta-lactamases that are normally reserved for multi-drug resistant infections and administered in hospitals. So for this reason, these are associated with hospital waste. And if you look at the bottom where it's partitioned into water or sediment, you can see that in the case of both classes of antibiotics, the sediment samples show similar trend, that downward trend, with the least amount of antibiotic resistance markers found in Sult and the most found in Toad Lagoon. Whereas in the water samples, you see it's more, the patterns are not coherent, and it's more site specific for each site, as well as the different classes. So what we're seeing overall is that in the case of the T4 samples, 2 weeks was sufficient exposure out in the open ocean alongside commercial oysters to allow for specific uptake of novel ARGs from the commercial oysters to the SPF oysters. And so what we resulted was site-specific profiles of the resistome with a high level of diversity and complexity. But while colistin resistance was common to all sites, most of the colistin resistance genes studied were colistin resistance genes that were intrinsic to the chromosome of the bacteria, and so they were not plasmid-borne and highly mobile, facilitating horizontal gene transfer. In the case of the Harvii bacteria that grew at 37 degrees, or I should say the Vibrio bacteria that grew at 37 degrees, most of those were from the Harvii clade, which contains a fairly large number of potential zoonotic Vibrio species. And so this may be something to look at in more detail in the future to increase surveillance of Vibrios found in oysters. We showed that there's geographic restriction among the different colistin resistance markers, and so for this reason, we advocating the restriction of live oyster transports, because in the past, it's already been shown that some oyster pathogens have been transmitted across international borders by the transport of live oysters. And also the early exposure to Nuflor was seen to be something that impacted oyster immunity, because in the Vibrio challenge that we performed, the oysters that were given Nuflor in the hatchery later, we're talking almost a year afterwards, when we did the bacterial challenge, the ones that received Nuflor had a higher level of survivorship when challenged with a specific oyster pathogen. In work packages 3 and 4, we saw that the sediments were influential in the uptake of antibiotic resistance genes by shellfish, and so this may be something to consider when choosing shellfish farming sites. We also saw that in the heavy metal contaminants seen in the waters at each site and in the oysters collected from each site, that there was specific heavy metal profiles from the distinct sites, and also that there was distinct pathobiome and rhizostome profiles isolated from each environmental compartment. In the case of the seawater, in particular, there's suggested sources for some of the drivers that are influencing antibiotic resistance uptake, and so this is something that may be worth looking to target specific sites for cleanup. So overall recommendation for Spare Sea is that we restrict live oyster transports and strictly control the use of antibiotics on the hatcheries where the oyster seed are produced. So if there are any questions that you'd like to ask, now is the time to ask them.
Yeah, Carl, thank you very much for this overview and taking us on board, let's say, how you arrived or derived, let's say, your policy recommendations. And well, are there any direct questions to Carl here from the audience? So if so, then please activate your camera and your mic and go ahead. Well, if this is not the case for the moment, then we move forward, and I can see that Andreas is already sharing his screen with us. So that means we move forward, let's say, to the second aspect. That's, let's say, about source and fate of AMR in aquatic ecosystems. And Andreas, supported by his colleague Claudia, will give us an insight into the findings behind this policy recommendation. Andreas, the floor is yours.
Okay, can you hear me?
Yes.
Yes, so thank you. It's a pleasure to provide an overview of our SARA project. In this project, we had many partners from different regions, starting in the north, Sweden, Germany, France, Spain, Portugal, Israel, Uganda, and Mozambique in Africa. And one of our objectives was to have really comparable data for the different regions. And so we started with method harmonization. This was mentioned already by Thomas in the introduction. So we put together the methods, and all partners analyzed the antibiotic resistance by cultivation in their own labs, and all applied the same method for the extraction of bacterial DNA and RNA. And then we could exchange the nucleic acid extracts, and the parameters in green were then analyzed by the specialized laboratories. So we really can compare the data that we produced, and the booklet summarizing the data is available if you're interested. So harmonization is very important. And also we have a good basis for selecting and recommendations of analytical parameters. Then we took samples from the model areas and the different partner regions. This is an example for our German area. And we have taken samples from the influent of the wastewater treatment plants and the effluent after treatment. And then we looked at the samples from the receiving surface waters, up gradient and down gradient of the wastewater influent. And we got the data here. You can see this is an example for antibioticides bacteria, ESBL bacteria, very high numbers in the wastewater, raw wastewater, then lower numbers in the effluent of the wastewater treatment plant, a little bit lower up gradient, a little bit higher down gradient, the wastewater influence. This is an example for Germany. We have taken these samples in all the different areas. This is a lot of data, too much to understand it easily. So we can focus on Sweden and Uganda. And so you can see for Sweden in general, the numbers are much lower as compared to Uganda, much higher numbers of antibiotic-resistant bacteria. And this is consistent with all the parameters. We have very slow prevalence of antibiotic-resistant bacteria in Sweden and very high in Uganda. This is for the cultivation method. If we look at the antibiotic-resistant genes, and these data are normalized with respect to the absolute biomass available in the different samples. And you can see by the different colors, low red means low abundance. Again, Sweden on the left here has very low prevalence of antibiotic resistances, lower as the other European partners and much lower as compared to the partners from Africa. So we can see, this is quite consistent in Sweden, low abundance, in Uganda, very high abundance. If we think about the reasons, in Sweden, we have already for a very long time strict regulations for the use of antibiotics, not only in human medicine, but also for food-producing animals. And we can see the results that we have lower antibiotic resistances. And in particular in Africa, there are not any regulations, so no restrictions. And we think this is one reason why we have such high resistances in Africa. Also, wastewater treatment is, of course, important. And here we have compared the effect of the treatment in Germany on the top and at the bottom in Uganda. You can see in Germany we have more than 4 log removal of the ESBL bacteria. In Uganda, only 2. We have different treatment systems. In Germany, we have activated sludge treatment. In Uganda, we have a wastewater stabilization pond that is less efficient. And if we look at the antibiotic resistance genes, we have a lower removal also in Germany, but still we have 2 log removal. In Uganda, we do not see any removal, or we even see an increase of antibiotic resistance genes in the effluent of the stabilization ponds. So this is a great difference. The reduction much lower in Uganda, and the stabilization pond there results in lower purification and even an increase of antibiotic resistant genes. So antibiotic resistances are then going to the environment with the wastewater treatment effluent. And we compared the stability of the different parameters in the surface waters. And you can see on the top, we have fast decay of ESBL E. coli and the corresponding antibiotic resistant genes. And we have a high persistency of the antibiotic resistant environmental bacteria that are analyzed by a different method, oligotrophic cultivation, and also the genes corresponding to the environmental bacteria are then much more persistent. So this means by introducing the resistances to the environment, we build an environmental reservoir. And as was already discussed before, there's a high risk that then the genes are transferred back to pathogens. That could be a real problem, and we should take care of the environmental reservoir. We are also interested, and we studied this in our project, what are the sources of the pollution that we observe in the surface waters in the environment. And so we have a lot of input from human pollution, but we also can have pollution from animal production like pigs or even from wildlife. And we can use specific bacteria and biomarkers to see what are the sources of the pollution. And in our project, we compared in particular the human markers. There are different markers that are published and that are used, and we were interested if these can be used in all the different regions from the north of Europe to the south of Africa. And we compared the results for the different human markers, and again, a lot of data. To make it short, the conclusion is that the crusphage, a specific human biomarker, was the most abundant marker, and it can really be recommended for human source tracking in the different regions and even other parts of the world. It was also studied by our partners in the project. So it's a very quick overview. So I come to the conclusions. Wastewater-based epidemiology is a very useful tool for pathogens monitoring. Here we have shown this for antibiotic resistances. Harmonized methods are available, can be used or really compare the data, and our methods are available on our project website. Antibiotic resistance is much more pronounced in Uganda as compared to Europe, and a lower relative abundance is observed in Sweden due to the many-year restrictions that are already applied. Wastewater treatment plant effluents are important sources of the antibiotic resistance. And during treatment, the bacteria are better removed than the resistant genes. Then we observe a faster decay of human-associated antibiotic resistances as compared to the environmental-associated resistant bacteria and genes. And the microbial source tracking of human markers, in particular of the crusphage, is suitable in Europe and Africa. And what I did not address, but we also have seen that extreme events can really lead to increased contamination and has to be considered in risk assessment. So as monitoring recommendations, I think the clinically relevant ESBL E. coli should be monitored. I think this is what many groups are doing already. We also should include the ESBL producing environmental bacteria because these reflect the environmental reservoir and we need this information. And of course, also antibiotic resistant genes should be included in the monitoring programs. I would like to thank all our partners, 8 partners. Here you can see the lead partners, contact person. And of course, I take the opportunity to thank the funding agencies for funding. In our German case, this is a German ministry, the BMBF, that just recently changed the name. So thank you very much for your attention.
Well, Andreas, thank you very much for this insights in the SARA project and those insights that helped us to elaborate on the recommendation regarding the monitoring. Are there any questions from the audience directly right now, for the moment, technical questions to Andreas before we get into the discussion later on? So if so, then please raise your hand, open your camera and your mic and go forward. Andreas, maybe a quick question from my side. You've shown, let's say, in the ponds in Uganda, the increase in the resistance
Resistance
genes. So do you have an explanation for the mechanisms?
I think in the ponds, the bacteria are in very close contact. Yeah, you have a lot of biofilms, and so these are the best conditions for horizontal gene transfer, and even some bacteria might grow there. We also have to add that these ponds are not maintained very efficiently, so it would be better if they would remove the solids more often. But these might be the reasons for increasing the genes. Yeah.
Right. Thank you very much for this. Okay, then we will move forward to the third insight on results that were that fed into the policy brief, and that will be given by Victor. And Victor, yes, we already see your presentation, and so the floor is yours.
Thank you, Thomas. Thank you very much, and good afternoon. So first of all, I would like to thank AquaticPollutants project for giving us the opportunity to present the Nature project results. So, well, Nature project stands for nature-based solutions to reduce antibiotics and antimicrobial resistance in aquatic ecosystems. And in fact, it addressed one of the topics of the aquatic pollutants call, which was the taking action, the point number 3. And it was produced thanks to the collaboration of 6 partners from different countries. First, CMR from Portugal, Aarhus University and Clean Water from Denmark. We have also in our consortium, Karlsruhe Institute of Technology from Germany, and also ABT from Mali. And finally, the SIG from Barcelona. So first, before starting with the project results, I want to explain you which is the current definition of nature-based solutions. So if you go to the European Commission, there is a definition, and I think it is quite good, which says that the nature-based solutions are in fact solutions that are inspired and supported by nature, which are cost-effective, simultaneously provide environmental, social, and economic benefits and help to build resilience. So here you have some examples of what nature-based solutions could be. For instance, constructed wetlands for treating urban wastewater. Here, as you can see, we can have also restored wetlands to treat or to remove pollutants from surface water bodies like rivers. We can have also buffer strips or riparian areas in agriculture to treat agricultural runoff. Or finally, what we can have is green areas in our cities to treat urban runoff. With this in mind, so the main objective of the Nature project was to provide evidence about the use of nature-based solutions for reducing antibiotics and ERGs at river catchment scale. So we are not going only to provide evidences that nature-based solutions can be a solution for treating wastewater at both at secondary wastewater treatment level and also at tertiary wastewater treatment level, in this case to produce reclaimed water, but also at river streams. So we can implement restored wetlands to deal with water pollution. We can renaturalize our river ecosystems. Or what we can do at the end, in our estuarine areas, we can use salt marshes to remove those pollutants that we have in our estuarine areas. For instance, to go with the results, so here we have some of the results from secondary wastewater treatment in Denmark, where we assess 3 different nature-based solutions, 3 different technologies for decentralized systems, for low populated areas, or less than 200 population equivalent. We assess 3 nature-based solutions configurations: vertical constructed wetland, aerated constructed wetland, and finally bioelectrochemically assisted constructed wetland for over 1 year. In all 3 cases, what we found was that the removal efficiency of antibiotics was high, around 90% for all antibiotics that we were able to monitor between the inlet and the outlet of each of those 3 nature-based solutions. Next step, we can go to use the nature-based solutions as a tertiary wastewater treatment to provide reclaimed water. And this is something that we did here in Spain. So in this case, we were assessing the use of nature-based solutions in centralized systems of treating 50,000 to 400,000 inhabitants. So here you have 2 configurations, the 2 nature-based solutions that we have assessed, a surface flow constructed wetland and an horizontal flow constructed wetland. And in this case, we compare the performance of nature-based solutions with the performance of a conventional tertiary wastewater treatment that we had in the area. In this case was sun filtration plus UV radiation and chlorination. And here are the results in summer and also in winter. So what we found was that the nature-based solutions were able to remove antibiotics, so in this case several antibiotics, from 80% to 90%. Whereas the reference technology, the most used technology for producing reclaimed water in Spain, is only able to remove between 30 to 50% of the antibiotics present in the wastewater treatment plant effluent. So once again, nature-based solutions can be or seem to be a good solution. Next step, so we can go to the streams and to the river streams. And if we go to Mediterranean countries, what you are going to find are streams which are dry almost all the year. And if you are able to see water, in most of the cases, it's going to be treated wastewater. So it means that what we have here is what we call wastewater effluent dominated streams. And here you have two of those streams in which the only water that flows to them is treated wastewater, in this case, from those two wastewater treatment plants. In one case, we have one stream which was completely covered by vegetation, as you can see here, and in the other case, we have a stream which has or had less vegetation, winter and summer. And here we have the results. So here is the total concentration of antibiotics at the wastewater treatment plant discharging point, and here is the stream from the wastewater treatment plant discharging point and downstream 1 kilometer, 2 kilometers, 3 kilometers, and 4 kilometers. What you are seeing here is that after only 1 kilometer downstream in the vegetated stream, in only 1 kilometer in summer, we are able to remove more than 80% of the antibiotics that were present in the wastewater treatment plant effluent. In winter, we were only able to achieve 40% removal. But if we go to the less vegetated stream, what we are going to see here is that the removal was less than 20%. Not only 1 kilometer downstream, 3 kilometers, and even 4 kilometers downstream, we didn't see much removal here. So once again, nature-based solutions are showing us the way to remove those antibiotics from the wastewater treatment plant effluents. And something similar was found in Denmark. Here, what we did was the assessment of a restored wetland in a river. This is the river, one of the rivers, Aarhus River. And here we have the restored wetland. And thanks to the restored wetland, we are able to remove more than 90% of the pharmaceuticals that we found at the entrance of the restored wetlands. Something similar was found in Portugal. So in Portugal, we compared one area with salt marshes and the other area which had less salt marshes. And what we found systematically was that after the salt marsh, the concentration of antibiotics, in this case clarithromycin or trimethoprim, decreased by more than 90% again. So once again, the presence of vegetation, the renaturalization of our river streams, our ecosystems, is improving the quality of the water. But not only about we are improving the quality of the water regarding to antibiotics, nature-based solutions are also affecting microbiological composition and antimicrobial resistance composition. So what we found was that the inlet at the entrance of the nature-based solutions, what we found was that the microbiological composition was wastewater-related bacteria, what we found. But at the outlet, we found river water or rhizosphere-related bacteria. So we were able to shift the microbial composition of the water, thing that was not able to be done thanks to the conventional tertiary wastewater treatment. So this is a good point for nature-based solutions. Not only changing the microbial composition, we are also removing antibiotic-resistant genes, as you can see here in this table. We were able to remove around 2 logarithmic units of antibiotic-resistant genes, thanks to the nature-based solutions. But in the reference technology, that removal was only around 0.2 or 0.5 logarithmic units. And finally, as a key conclusions of the nature project, what we found is that antibiotics and ERTs have been found in wastewater, reclaimed water, river streams, but also in estuarine areas. But thanks to the nature-based solutions, we demonstrated that we were able to effectively remove those contaminants, antibiotics, and ERTs from wastewater and also from river streams and estuarine areas, in this case by 99%. And finally, and I think it's very important, nature-based solutions were able to shift the microbial community's composition from fecal-associated bacteria at the inlet to river-related bacteria at the outlet. As a key recommendations of the Nature project, what we can say is that we can promote the use of nature-based solutions, such as constricted wetlands or riparian buffer zones, to effectively reduce antibiotics and ERGs from wastewater treatment plant effluents, and also agricultural runoff before they reach water bodies. So it means we can use nature-based solutions at source to avoid the entrance of those pollutants in the water bodies. So, but for doing that, we need to recognize that nature-based solutions are, in fact, environmentally sustainable approach to mitigate AMR and contamination, provide funding for pilot projects across diverse European regions to evaluate the effectiveness of nature-based solutions in different contexts. I think it's very important just to provide evidences not only from our case studies, so I think we need to get more information, to provide information to all the stakeholders and all policymakers to understand that nature-based solutions can deal or can be a solution, and support broader adoption of nature-based solutions to help reduce the spread of AMR in aquatic environments. And finally, I always end my presentation with this sentence, which tries to summarize what we have done in the Nature project, which is bringing nature back into our ecosystems for addressing the new challenges stemming from human activities. Today we are talking about AMR. Tomorrow maybe we can talk about microplastics or other pollutants that may be a problem. So finally, I would like to thank all national funding agencies, and also if you want to find more information about the project, you can check our website, or you can follow us on X. So thank you very much.
Well, Victor, thank you very much for giving us the insights in the Nature project and your nature-based solutions. And again, also here, so the quick question, are there any direct questions to Victor at this point? Well, if this is not the case, then we can come to the, let's say, to the final discussion round. So, of course, let's say we have prepared some questions we would like to discuss with the speakers. Of course, for you as the audience, your, let's say, our target audience. So, are there any generic points related to the presentations, the policy brief, things you would like to address here in the discussion before we get into the discussion round with the group? Well, if this is not the case, then we'll get into the panel discussion round, and whenever you see any question or issue arising from your side, then please feel free to get back to me into the discussion, then we can start with that. So I would like to start with a question related to the, let's say, aquaculture environments that's related then to Carl. Carl, let's say in the SPACI work you have described, let's say, WWTA genes that are widely present in Vibrios. Do you see any kind of risk that's coming out of these resistance genes that could be present for human health? And maybe a question that's also related to all three speakers. When we compare the AMR and ARG issue, let's say, with pollutants, is there a kind of a risk that, let's say, new resistance genes occur that we need to take into consideration separately to have the whole, let's say, assessment procedure and everything running through for them as well? Or can we say, let's say, from the environmental behavior, from the risk things, are they comparable? But Carl, maybe first answer to the first part of my question from your side.
Well, the the EPTA, I should maybe explain for people not familiar. EPTA is ethanolamine phosphotransferase. It's a mechanism of modification of the cell lipid membrane, the LPS of the membrane of bacteria, particularly gram-negative bacteria. And the MCR, it encodes for a similar enzyme performing the same function. The difference between MCR and EPTA is that MCR is a mobile enzyme. It's a gene that's carried on a plasmid, and it's been a source of worry for many parts of the world, particularly Southeast Asia, where this type of colistin resistance has spread widely and is a real problem. EPTA, on the other hand, it's not encoded on a plasmid. There are, among the EPTA variants that we studied, the majority of them were genes that were intrinsic to the chromosome of the bacteria. They're not so highly mobile. Among the EPTA variants we looked at, very low percent, I think maybe it was 5% or something like that, they were associated in the chromosome of the bacteria with mobile genetic elements. So there's a very low possibility of them being mobilized and creating horizontal gene transfer that could create novel pathogens. However, that being said, if a zoonotic vibrio containing one of these intrinsic EPTA genes in their chromosome were to infect humans, that would be a health risk. But since most of the bacteria that we're talking about are coming from a marine environment where the temperature is much lower than 37 degrees, the majority of the bacteria aren't going to represent zoonotic risks. I hope that answers your question with some qualifiers.
Yeah, right. Carl, yeah, thank you very much. And maybe the second part is maybe something that, I don't know, maybe Andreas could also provide an answer for. It's like, let's say, when we look outside, let's say, into the aquatic environment, and we see a status at a certain point with a certain set of, let's say, resistancy genes being there, being present. Let's say how dynamic is this system? How fast is this community, or let's say the pattern of genes changing with respect, let's say, related to the change of, let's say, human activities, or let's say the use of antibiotics and things like that. What I would like to know is, let's say, are we here, let's say, caught in a kind of a cycle that we always have to look for new ARGs coming up, assess them to see are there potential risks coming up out of them, or how do you assess that? Maybe Andreas, maybe you might be able to say something on that. Maybe you already had some information on that gathered in the SARA project.
I think it's good to ask this question because if a new gene, this gene is coming up, I think we have to study this gene. And as Carl mentioned, mobility is quite important to understand. Of course, you can check if it's more located on plasmids or mobile elements, but I think in general we need some more knowledge and studies into the gene transfer process, in particular between environmental bacteria and pathogens. I think that would be very important. So I think we can conclude some general rules from what we have seen during wastewater treatment, for example, that we can remove all of these genes by treatment, like ozonation, for example, what we have done in other projects. But to understand the behavior and persistence in the environment, I think this is something that has to be studied specifically, for the parameter we are interested in.
Okay, but that clearly means due to that, even let's say the issue of, let's say, harmonized environmental AMR monitoring standards, so the monitoring is even more crucial, more important to be aware to see also if new things are coming up, and to be upfront to potential risks occurring out of these resistance genes, right?
That is my opinion, yes. And so far, most of the groups focus on the clinical relevant antibiotic-resistant bacteria. I think this is most important, of course, but we should have more insights into the fate of the environmental antibiotic-resistant bacteria. And so we suggest to include this parameter in the monitoring to get more information over time. Yeah, I think this is really important. Yeah.
You raise a good point, Andreas, that the antibiotic resistance of non-clinically relevant bacteria, given that there is horizontal gene transfer occurring in various modalities, environmental bacteria containing antibiotic resistance genes of high risk, like colistin or extended spectrum beta-lactamases, they're equally important, and there should be monitoring for them as well. I was curious in your presentation when you were talking about traceability, you showed these tracing factors related to human, but do you have a similar slide that shows this for the environmental and animal related bacteria or antibiotic resistances rather?
Yeah, we have done this in other projects where we looked at the sources for pollution, including antibiotic resistances, and there you can see that this can be related to surface runoff from chicken farms, for example.
Mm-hmm.
Yeah. And then you can, if you don't know exactly in your area what are the sources of pollution, then microbial source tracking really can help. In this specific case, our conclusion was that most of the pollution is coming from a retention basin, for heavy rain events, and this was too small. So we could make the very exact recommendation to increase the volume of this retention basin, and this really helped. Yeah, because in the beginning we didn't know what is the reason for the pollution.
Yeah, and you I think advocated for increased monitoring after specific environmental events, something to that effect, yes?
Yeah, this is what we have done. This is published already. We can send
it.
Yeah.
Likewise here, after a very large rainstorm that caused a tremendous amount of flooding and property damage and a tremendous amount of terrestrial runoff into the bay here where they grow the oysters, we saw large increases in some of the heavy metals that we were tracing. Cobalt, manganese, zinc, all increased a lot after this big storm event. So it is quite clear that what you say is true, that the monitoring maybe should be augmented when there are major events like that.
Yeah, but that brings me to the point that all, let's say, leads into that besides the monitoring, we need to take action. And we had, let's say, what we have in our policy briefs, so the recommendations also for the nature-based solutions. And Victor, having said this, then also when we see the issues that Andreas has mentioned, that Carl has mentioned, and where we see that we need to foster the resilience also for our water systems, and that in the various, let's say, services that these aquatic ecosystems provide for us, being it, let's say, for the aquaculture stuff, being it, let's say, for the aquatic ecosystem as such, being as a drinking water resource, or being for agriculture. So what do you see? Let's say we have seen that in the actual, let's say, draft of the European Water Resilience Strategy, the nature-based solutions play a quite dominant role. Can you elaborate a little bit more on that why, from your perspective, you think that nature-based solutions are so important and can achieve a real big impact, how they could really contribute to more resilient management of water from your perspective?
Well, thanks, Thomas. So nature-based solutions, I think they can contribute to more resilient water management. So by using natural ecosystems. So we are using what is already in the nature. We are using, in this case, wetlands, river banks, buffer strips, forests, and renewable species to deal with this problem, to reduce flood risk, to improve water quality, enhance groundwater recharge, and maintain ecological balance. But at the same time, which is good from natural-based solutions, is that we can offer a cost-effective alternative to traditional gray infrastructures that adapt to climate change. We can support biodiversity and provide social and economic co-benefits to communities. And I think this is very important and relevant from the point of view of citizens. And this is something that natural-based solutions can provide.
I have a question for Victor. It was interesting to see in your presentation how the linear distance of the drainage canals was almost as important as the amount of vegetation. And I was curious in knowing if perhaps there's a way to sort of codify how to design these kinds of drainage channels. If you're going to design I mean, some of these canals are natural, but in the case where you're going to design a canal, like you have a canal that's coming from a wastewater treatment plant and currently is just a concrete conduit, and you want to convert at least portion of that to vegetation to help clean up things further. Is there like a minimum linear number of meters or perhaps kilometers that you think could be recommended for minimizing the amount of antibiotic residue?
Yeah, I think the first thing is the renaturalization of the systems. So to bring back ecosystem nature to the streams. And the other one is to include meanders. So we need to include meanders. It cannot be a linear canal. It should be a meander canal and with vegetation. And we need to increase the retention time and the interaction of the water with the plants and the vegetation. So we
Okay,
need
this is what the meanders serve, is to increase retention time and increase reaction with the plant vegetation along the shore.
Exactly. So we need to provide something like that.
Right. Thank you very much. Let's say with having all these things in mind, so that brings me back again to the monitoring thing. Andreas, you mentioned, well, you already have elaborated in the SARA project. You have your booklet on the harmonized methods. Do you see a way forward for that, bringing this also, let's say, in a standardization environment? Would it make sense to have or to bring these developments forward, for example, to elaborate a kind of a SEN standard or ISO standard around that, or are there already activities underway that we can expect in the maybe in the near or the mid future, already standardized approaches on there? So that would be interesting to see what your perspective is around that. Is there any kind of standardization activities needed on that, or would they make sense?
Yeah, from my point of view, we need some standards and some harmonized methods, because now we have the antibiotic resistance that are also considered in the new EU regulations for wastewater treatment. It's not defined yet, but we need a discussion which parameters are suitable for monitoring, so we can give some input from our project, but we have to discuss with other colleagues. And I think then the community has to agree on some parameters that are suitable for monitoring, including bacteria. I think this should be clinically relevant, but also environmental bacteria. And we have to discuss which antibiotic resistant genes should be monitored. And then we have to discuss which method in detail should be applied. In case of quantitative PCR, which primers should it be conventional short amplicon or probably long amplicon PCR? So still many things to discuss. But standardization will be important for developing the regulations.
Yeah. Okay, so that's really then crucial also with what we said with the harmonized and integrated surveillance framework, and to bring this really forward.
Mm.
Right, thank you very much for this. So I need to go back to you, Vic. Yeah, Louis, there's your hand up. So then please open your mic and finally also the cam, and yeah, please go ahead.
Hello, good afternoon. My name is Luis. I'm from Spain. And well, I just wanted to say that I work in the Joint Action for Antimicrobial Resistance and Healthcare Associated Infections. And this is a public health project at the European level that started last year and will run until end of 2027. And it's involved with the human sector, the animal sector, and the environmental sector. And we are currently working with ministers of health and other environmental authorities in Europe to work on standardization of AMR indicators for environmental monitoring. We are approaching most feasibly the wastewater compartment, but there are other compartments to discuss. And we have launched a survey to agree on indicators to have a first level of evidence to start gathering protocols from countries and having a first proof of concept of where the direction is going to go. So I will put in the link the surveys, and we will let you know about the next activities so that you can also join us in the decision making.
Yeah, Louis, thank you very much. I think this is very valuable and helpful. I would like to go, let's see, with respect to the time, to last question that I would need to bring me back again quickly to Victor. So when we all are aware, let's say, we've seen that nature-based solutions have an important role in let's say, reducing the spreading of AMRs. But let's say we are all aware that we also have a kind of space requirements for AMR solutions. So you've shown the various options where to have it in agriculture, surface water areas, in the urban sector, in the agricultural sector. Do you see any kind of progress, or what are the perspectives to cope with this space demand of this thing? Is something we could get around? We could have solutions for?
Yeah, this is correct. So, but we also need to consider the two additional aspects to this drawback. So the first one is the co-benefits of nature-based solutions compared to gray treatments, such as the provision of ecosystem services and other beneficial, including climate change mitigation, of course. And the second point, which is important to take into consideration, is the existence of what we call intensified nature-based solutions. So this is still room for improvement here. They are nature-based solutions designed to enhance pollutant removal while requiring less surface area, which is one of the main drawbacks of nature-based solutions. For example, biochar-based coastal wetlands for wastewater treatment, or biochar-based buffer strips for treating agricultural runoff. So preliminary results from those systems showed that intensified nature-based solutions can be indeed the solution to reduce the main drawback that always nature-based have had, which is the large surface area demand.
Yeah, all right. Well, Victor, thank you very much for this. Well, with respect to the time, I would say we need to come to an end. So let me quickly wrap up. So I think what the three of you has presented out of the projects, what we have used, let's say, to formulate it around some key recommendations around the policy sector is quite important. We'll move that forward. We'll for sure have some impact. We've already seen that there are routes forward, let's say, from the nature-based solutions with respect also to the water resilience strategy. We've seen for the monitoring stuff, for the harmonization and standardization things, there are routes moving forward. And we've also seen, let's say, for the aquaculture sector, that we have issues going on there that we need to take action to take care of this very important route in terms of spreading of resistance genes. So plenty of things that we can bring forward. And let's say, besides the issues around the policy brief, I've mentioned at the beginning that at AquaticPollutants, we also were looking to various stakeholders. And of course, let's say policymakers is one group of them. So we have plenty of them in there. That's also why, and this is what I would like to use at the end, why we have let's say, elaborated in the project, also additional information material that I would like to share the information with you. That's on the one hand side. So we've elaborated a layman's report that's available on the AquaticPollutants website that provides some kind of easy information that's also could be important for you as the projects to work with it, but also, let's say, for stakeholders and end users. And we have our Transnet viewer where we, let's say, bring together knowledge demands and knowledge out of the projects so that they can find their way into practice. And of course, let's say what comes next. We will now finalize our policy brief in the coming days. So that means at the latest from mid-June, you will find the final version of the policy brief at our website. That's then also available for you. Yeah, well, and with this, I would say it's up to me to say thank you very much to all of you for being with us, especially to Andreas, Carl, and Victor for giving us insights into their work and supporting us also within the setup of the policy brief by providing so excellent work out of their projects, together with the results of other projects that's also feed into our four, let's say, key messages from the policy brief. Well, and with this, I would like to say thank you very much to all of you for joining us today. And with this, I'd like to say goodbye and have a very nice rest of the day. Thank you very much.
Thank
Bye.
you for hosting, Thomas.
Thank you.
Bye now.
Bye. Bye.
Bye.